A very good morning, students. This is your teacher Neelam Helawat, and I hope you are enjoying your studies while sitting at home. Isn't it? Right, everyone. So we'll continue with the topic, that is your genetic code, and this is your part number 2. Yesterday we have spoken about a general introduction regarding the genetic code, right? Now today we'll be talking about the features of the genetic code. Now see, I told you that the genetic code is a triplet one, isn't it? So the first point will remain the same. The code is a triplet. Triplet means that it is composed of 3 bases. Like you can have A, G, C, isn't it? You can have G, T, A. So it is basically made up of, the code is made up of 3 bases. That is why the code is a triplet. And there are in total 64 combinations possible. As I said, 64 codons, right? This also we have discussed. So there are in total 64 codons, and out of these 64 codons, 61 are your sense codons. They will be coding for the amino acids, while 3 codons, they do not have any function or they do not code for amino acids. They only stop the process, and that is why these 3 they are called as stop codons. Okay, they're called as stop codons. So let's see, let's again we write it. So in total we have 64 codons, right? And out of 64, 61 are will be coding for your amino acids, and 3 codons, they will act as stop codons. Okay, they will act as stop codons. And your triplet, the codons, they are in the form of 3 bases, so they are triplet. The second most important feature regarding the genetic code is that it is a degenerate. Now, what do you mean by the degenerate? This means one amino acid can be coded by more than one codon. Okay, is it clear, everyone? Just have an example of this. Let's take an example of this. Suppose there is a proline. So proline is an amino acid, right? So proline is an amino acid. It has 4 codons. These 4 codons, they together code for the proline only. Okay. So this is an example that an amino acid can be coded by more than one codon. CCU also code for the proline. CC also code for proline. CCA also code for proline. And CCG can also code for the proline. So 4 codons, they are coding for the one amino acid which is called proline. Okay, dear students? So I hope this is understood. Right? So your amino acids, they are coded by more than one codon. For example, the proline. Proline is coded by 4 codons. Okay, so that is why we call it as degenerate. Okay, the genetic code is degenerate. Let's revise it again. Codon is a triplet. There are 64 codons in total. 61 codons, they code for amino acids. 3 codons, they are stop codons. They will stop the process. And amino acids, they are one amino acid can be coded by more than one codon. So this is called degenerate. I hope it is clear, everyone. Now codons, they are always read in mRNA. See, I told you, you will have DNA, isn't it? So from DNA, mRNA will be made. And this mRNA has the information to code for a specific amino acid. Okay, clear? Now this A, G, T, C, A, G, right? TA, this sequence is continuous. There is no gap here. So in mRNA, these bases, they are attached continuously. So there are no comma, there is no punctuations in between. These base pairs, they are arranged in continuous fashion. Right, everyone? They are arranged in continuous fashion. Next is the code is universal. The code is universal means, suppose I say AUG. Remember you? You remember, I think? AUG is your initiation codon and it codes for amino acid methionine, isn't it? It codes for the amino acid methionine. So AUG, wherever it will be present, it can be the bacteria, it can be the human beings, always the AUG will code for methionine, right? Similarly, we can have an example of triple U. Now this triple U will code for phenylalanine, right? It will code for phenylalanine. So triple U will always code for phenylalanine. Whether these three codons, or you can say whether this three nitrogen bases, they are present in bacteria, they are present in human beings, or they are present anywhere, right? So AUG will always code for methionine, triple U will always code for phenylalanine. So that is why we say this code is universal. Okay, dear students, this code is universal. Let's revise again the third and fourth point. First of all, the bases, they are arranged continuously. There is no comma, there is no punctuation. Second is, this code is universal, right? Means every codon will code for the same amino acid, whether it is present in bacteria, whether it is present in human beings, whether it is present in prokaryotes, whether it is present in eukaryotes. That is why we call it as this code is universal. But there is a few exceptions that have been found in mitochondrial codons. Okay, dear students, there are few exceptions that have been found in case of mitochondrial codons. There are exceptions in case of mitochondrial codons. Okay, rest it will be followed everywhere. Now second is AUG has dual function. Just now we discussed and we have spoken in earlier videos also that AUG is your start codon. Okay, AUG is your start codon. And secondly, it will form an amino acid methionine, right? So AUG has dual functions, means it performs two functions. First, it acts as a starting codon, and secondly, it will code for amino acid methionine, right? The three codons which do not produce any amino acids, they are called stop codons or terminator codons. They are called as stop codons or terminator codons. They are UAA, UAG and UGA. Clear, everyone? So 6 important functions, or you can say 6 salient features, the characteristics of the genetic code. What is the first one? That there are 64 codons, 61 will code for amino acids, 3 will be acting as stop codon. Secondly, the code is degenerate. Third, the code is universal. Fourth, what is the fourth one? There are no punctuation. The sequence is continuous in mRNA. Then AUG has dual functions and 3 codons, they will act as stop codons. Okay. Now this is your table. This table is very, very important, students. You must read this, and I'll tell you the ways to remember it. See, this table, first of all, we have written the 4 bases. See, we have only 4 bases: U, C, A and G. So we'll be writing here also, on this side also, and this side also. So we have written bases on 3 sides. First on this side, then here, right? Clear? Now we'll take the first letter from this side, second letter from this side, and third letter from this side. Okay, dear students, we'll be taking the first letter from this side, second from this side, and third from this side. Okay. Okay, so first codon will be taking from this side. Suppose here it is U. So we'll take first in this block, first block I'm talking about. This is your first block, isn't it? This is your first block. Okay, so here we will have first letter here, this U, then second this, and third this. Okay, so that is why we have triple U. Clear? Second. Now second we will have this first letter, and this will have the second letter from here. So U, U, C. Third, first and third, that is A. Then fourth, U, U, this will remain same, we'll have this U. So we have 4 codons, okay, triple U. In the first box, we have 4 codons, triple U, then WC, WA, and WG. The first two, they code for phenylalanine, right? They code for phenylalanine, while the second two code for the leucine. Okay, second two code for the leucine. Clear? Let's take the second one. Let's talk about the second block. So we'll take the first letter from this side, second from this, and third from this side. So C, U, U. Okay, this one. C, यहाँ से first, and this. So C, U, U. Then C, U, C. C, U, A. Then C, U, G. Okay, so in this manner we will be making the complete blocks, and we can make 64 codons very easily. Okay, dear students, we can make how many? 64 codons. And just look at this. All these four, they are making the leucine. So we have done two blocks so far. So 4 plus 2, 6 codons, they are coding for leucine. Just now we have done 6 codons, they are coding for the leucine. Now if you look at the table, we see in these two blocks only one base is different, isn't it? Rest two, front two are the same. Here in first table, these two are same, these two are same, these two are same, these two are. Second also, the first two member is same, right? Only the third is different, isn't it? The third portion is different, and this has come from the Vobel hypothesis. This has come from the Vobel hypothesis. There was one scientist who proposed the Vobel hypothesis. Let's read about him also. Now, Vobel hypothesis. It says that the codons base pairs are in antiferal fashion with the anticodons on tRNA. See, if this is your mRNA, on mRNA you will have the codons, right? Then this will be your tRNA. This is your tRNA, right? So I hope you remember that codon is present on our mRNA, and you will have anticodons here, right? And these anticodons, they will pair with the codons, and correspondingly the specific amino acid will be formed. So anticodons, they are present on tRNA, and codons, they are present on mRNA. Clear? Is it clear? So anticodons, they have always the complementary sequence to that of the codon. Now this Vobel hypothesis, it says the rule focuses that out of, if there is suppose A, U, U, there are 3, now this is a codon. In the codon, there are 3 nitrogen bases, right? One is A, second is U, another is U, right? This says that base pairs are relaxed at the third position, so that a base can pair with more than one complementary base. Now, these two should be same. Any two out of this should be same, and third could be relaxed. We can have a different nucleotide base at the third position. Like you can have AUG. You can have AUA. So third can be different, right? So Vobel hypothesis says that the base pairing is relaxed at the third position, so that a base can pair with more than one complementary base. Some tRNA anticodons have iosine at the third position, right? And this iosine can pair with U, it can pair with C, it can pair with A. This means that if we have 61 sense codons, or 61 codons coding for the amino acid, right? So we do not require 61 tRNAs. Okay, देखिए हमारे पास 61 अमाइनो एसिड्स बनाए जाते हैं, ठीक है? इसका मतलब है हमारे पास 61 एंटीकोडोन्स होने चाहिए जो टीआरएनए पर मौजूद होंगे. लेकिन वोपल हाइपोथेसिस ये कहता है कि टीआरएनए में ज़रूरी नहीं है कि हमारे पास 61 टीआरएनए होने चाहिए, बिकॉज आयोसिन अगर मौजूद है, आयोसिन एंटी कोडॉन में मौजूद है, तो वो यू, सी और ए किसी के साथ भी पेयर हो सकता है. सो we require very less number of tRNAs to produce 61 amino acids. This is what the Wobble hypothesis says. Again see, mRNA पर कोडोन मौजूद होता है, tRNA पर एंटी कोडोन होता है. ठीक है, तो अगर हमें 61 अमाइनो एसिड्स बनाने हैं तो इसका मतलब है कि that we will require 61 tRNAs. But the Wobble hypothesis says that it is not necessary. Anticodons can be relaxed at third position, right? They can be relaxed in third position. Now, first two bases in the codon in mRNA, they pair traditionally with the second and third base of the anticodon, right? अगर हमारे पास AUG है, सपोज करिए ये हमारे पास एक कोडोन है, ठीक है, तो इसके जो दो बेसिस हैं, जो फर्स्ट टू बेसिस हैं, ये 100% tRNA के सेकेंड और थर्ड बेस पेयर के साथ पेयर होने चाहिए ट्रेडिशनली. ठीक है, सो दिस फर्स्ट एंड सेकेंड विल बी पेयर्ड विद सेकेंड एंड थर्ड. ओके, दिस इज कॉल्ड अ ट्रेडिशनल पेयरिंग. दिस इज कॉल्ड अ ट्रेडिशनल पेयरिंग. जब mRNA के कोडोन्स tRNA के एंटी कोडोन्स के सेकेंड और थर्ड पोजीशन से पेयर हो जाएंगे, वी कॉल इट एज ट्रेडिशनल पेयरिंग. ओके, बट थर्ड, रिगार्डिंग द थर्ड बेस, देर इज नो ट्रेडिशनल पेयरिंग दैट हैज बीन ऑब्जर्व्ड. सो दो बेसिस की ट्रेडिशनल पेयरिंग होती है, वो 100% कॉम्प्लीमेंट्री बेस के साथ पेयर होंगे, लेकिन थर्ड न्यूक्लियोटाइड, थर्ड बेस की कोई कॉम्प्लीमेंट्री पेयरिंग नहीं है. राइट? क्लियर है? एंड क्योंकि ये थर्ड बेस पे ट्रेडिशनली पेयर नहीं होता है, दिस इज रिस्पॉन्सिबल फॉर वूबलिंग. राइट? दैट इज वाई वी हैव वूबल फिनोमेना. क्लियर है? सो mRNA पर दो बेसिस जो मौजूद हैं, वो लाइक फर्स्ट एंड सेकेंड, दे विल बी पेयर्ड विद सेकेंड एंड थर्ड ऑफ tRNA. The third base pair does not have necessarily the complementary sequence on tRNA. So this is not a traditional pairing, right? Because of this, the wobbling will occur. Okay? As I've discussed it also. Now suppose this is your sequence, right? 3-3, and there is no comma, there is universal. Now try to find, suppose this is there on mRNA, this sequence is there on mRNA. Now try to find the anticodons on tRNA. चलिए anticodons पता करते हैं. tRNA पर इसके anticodons क्या होंगे? We know A can pair with T, U can pair with G, and G can pair with the U, isn't it? So two base pairs, they are 100% complementary. A will pair with T, U will pair with G. But third, there is no necessity that it always pair with U. This is no guarantee. Okay? But yes, we can generate a sequence based on this easily. U is G, G, isn't it? We can propose a sequence on tRNA, right? So it is very easy because mRNA is there. So complementary sequences will be there on the tRNA, which will have the complementary base pairs on tRNA, right? Is it clear? So we can easily project the base pairs which will be present on tRNA, right? So that the pairing can occur. Now just imagine, suppose I give you this sequence: met, methionine, phenylalanine, 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 phenylalanine. Now can you easily project the sequence of base pairs? Tell me the sequence of base pairs. Now in this case, just think what will happen. Is it easy or difficult? It is an easy or difficult? See, phenylalanine, if you remember, we've talked about the sequences. If you remember, just see this. Phenylalanine is coded by 3 base pairs, isn't it? It is coded, it can be triple U, it can be WC, it can be. So we are not sure that which base will be there, which codon will be there, isn't it? Yes or no? So we don't know which codon will be there. Now for methionine I can say, that it is AUG. But what about the phenyl? Which codon is present? I am not 100% sure. So if I have been given the amino acids, I cannot predict the codons with 100% surety. I may try, but I cannot give the 100% surety. But in this case, we can give, isn't it? Because the base pairs will always be complementary to the bases present on mRNA. Clear? We can say that the bases will always be complementary to that of the mRNA. Okay, dear students? I hope you've understood it. So अगर हमें अमाइनो एसिड्स गिवन है तो हम सीक्वेंस 100% गारंटी के साथ प्रोजेक्ट नहीं कर सकते हैं, लेकिन अगर हमें बेसिस गिवन है तो हम श्योरिटी के साथ कॉम्प्लीमेंट्री बेस पेयर्स बता सकते हैं कि कौन से बेसिस टीआरएनए पर मौजूद होंगे. Clear everyone? Okay, so your homework for today is make notes of this topic. Okay, dear students, make notes of the topic. Right? So today you will make notes of this topic. And secondly, since we have completed the code, genetic code, you will complete this topic, right? And you will attempt NCERT questions related to this. So we will complete the NCERT questions related to the genetic code. Okay, everyone? Is it okay? So you have to make notes of the topic, and secondly, you will complete the NCERT questions, okay, of this topic, genetic code. So, okay, everyone, that's all from my side for today. Goodbye, everyone.